So welcome to this section on bioassays or packages as part of the course F1 on flow cytometry and analysis. So similarly to ggplate, this part is looking at how we can use molecular data to be visualized as an assay using a range of functions associated with that package. So this package works well across different plate sizes. So from 6 well, 12 well, 24, 96, 384 well plates, all we need is to input our values observed, and then the metadata associated with that, and we've imported that in CSV format to be able to visualize wells like those shown here. There's a few functions we need to think about firstly. So we may have metadata embedded inside the actual file names themselves, and we use this function extract underscore file name to extract that information. Then there's a function data to plate format, which formats the plate data into a matrix with the rows and column names corresponding to that data. So we'd send the file name from here to there. So for example, we can take in the example data for this package called raw data 24, specifying the plate type corresponding to that, and then that will be imported in here into the into an appropriate matrix. And we can change the plate type depending on what we want. This then allows us to use the plate to df function, which converts that matrix into a data frame. So a data frame is a type of table, so it's specified rows and columns for more easy manipulation. So that can mean that we can import our raw data into the data to plate format and then send that matrix down to the plate to df function. So be able to look at the information in that. So what we would see is the rows and then the columns. So we might have A, 1 to 12, B, 1 to 12, and so on. The position is specified, and then we have the value we observe. So it's a lot like the ggplate package. Then we have a function known as matrix 96, which converts number of character columns into a matrix. It understands the plate type and source and can convert negative or NA values to 0. However, you should note that it's only available for 96 well plates. It's not available for the other plate sizes. So what we can do is take on our metadata file and send it to that function and use the ID inside that metadata file, example 1 here, to extract that metadata. So what we would see then is metadata, so from A to H in this case, and 1 to 12 for the different things that are labels inside this different metadata. So what we could do then is link these functions together. So we have the data matrix that's imported to plate to df, which is imported as a matrix. And then we can visualize our data like this. So we have A to H and 1 to 12 shown across there. So this is kind of our plate values that we want to look at further. We can then link in our plate metadata function, which combines plate-specific information. So that could be serial dilutions or something like that. And that function is more complex because we might have certain details embedded in there depending on what we're looking at. So you might have the ranges going from 1 to 8 here, the concentration we use for that serial dilution, the compound and things like that, whatever metadata is useful for us. I note that it's a list format, so that's why we have a kind of a list entry corresponding to this descriptor here, taxol. The same for concentration and that vector. So that list is then read in by the plate metadata function, as well as the metadata, and we're merging it by type. So you can see there we have a type column here specified in our plate details, which is also going to be in our plate meta file 96 as well. So to make sure that those column names correspond to each other, so they have to be exact matches, lowercase type in this case. And that means we can look at the structure of the plate metadata, which contains the row and rows and columns, the type as up here, S1, S2, S3, the position like A01, the ID, the dilution level, the concentration, which we see above, and then the compound type as well. And we can look at the head of the metadata as well. So you can see here the rows, A, B, C, D, whatever columns, the type, which is the metadata, the position, which is the well, the ID, the dilution level, the concentration, and then the compound as per relevant. We can also use the heat map function to visualize this data. So we take import our metadata as a matrix, specifying the plate type and the size here. So that plate 1 is the title. So we'll see a picture like this where we have our blanks and samples and standards visualized here across our different plates. We can also use the data as well as the metadata. So we can import the 384 plate as a matrix with this title of interest, and we just toggle the size to be smaller to visualize this full plate. And then obviously the next step will be to link in the raw data, the raw values data, in as a data frame D1, as well as the metadata. So if the metadata is being joined with D1, and you see that it's by row and column and position. So those corresponding column names that we had before here, we have row, col, and position have to be in both files, and they have to be with those exact labels. So you can change it to a different names, but they have to correspond between the different data names. And we're using here is the dplyr function inner join to join these together. So we're saying by row, column, and position. So it's saying basically where we have a match between D1 and the meta file, where the row and the column and position matches between the different files. We're going to join them together and put them in a new data frame D2. And this is using the dplyr package, which is why we have the 2 colons there that we're using this inner join function from that package. And what we'll see then is this kind of representation at the end. We have a row, column, position, which are specified up here. The position, sorry, the value, the cell, the compound, concentration, type, and the dilution. So you can see that effectively what we did, if you can think about it, is the metadata values have been imported into our plate here. We've also got a function called reduce blank, which might be useful if you have blank values. So that can infer missing data in effect for those blanks. So I take D2, which we created already, so that's this table here, which contained certain blanks inside it. And we have our drugs of interest and then the blank values. And it's going to get the mean of the blank values for drug 1, 2, 3, 4, and so on. And then we can view it. And what we'll see then is the row, column, position, the values we have there. And we see this is for the blank 1 here at the top. If we look at the histogram of the inferred values, so we have this new column called blank minus that we can put into blank. And when we go through the R code, we can look at this in more detail. We can plot it with a pseudo number of breaks. We can see that the average effect of that blank is 0. So although some of the data are spread around 0, the net average effect of the blanks is going to be 0. So it's kind of inferring the more accurate effects of the blanks corresponding to the different drugs that have been used. So moving on, then we have more functions beyond this reduce blanks. So we have df summary, but that takes in multiple data sets or groups, summarizes data, and so on. This is a more complex function. So if we take in our D2 matrix we had before, we put that as our first item here for df summary and send that to result 2. So we're looking at the values. We have a optional nickname, which is up to you. We can not remove rows with empty values. Then we have the blanks corresponding to what we had before. We have the other data that's in there, the cell, compound, concentration, and type. And then we have a cutoff thresholds as well. So what we're doing is summarizing the value after grouping the samples and omitting the blanks. This is the order by which the table will be arranged. So the strings above must strictly correspond to the entries in your data. So these have to be exact matches. So if we look at the arrangement of this, we have the cell, the compound, the concentrations, the type, the label, which is the nickname we had before, the number of observations, the mean standard deviation, and critical value as well. So what we can see then, if we look at this further, is that we have this convenient function called p-value, which applies a t-test comparing result 2 against the reference of the C1 values. So if you look here, we have certain values that correspond to this category of C1. So it's comparing these other values in C2 and C3 against that C1 group. So you can set this up depending on what way you want it to be. What we can do is import that t-test result into res1, which contains 24 rows altogether, so 24 tests, and it has 10 variables inside there. So what we can do is, some of our p-values are written like this. Something effectively, it has this portion on the right-hand side, less than 0.001 or something like that inside here. So what we want to do is get rid of the space and the less than symbol, so we're doing numerical values only, because we should never use rounded p-values. We should always use exact p-values for reporting our data. What we're then selecting are the p-values less than 0.01, because there are some values that are NA because they're the controls, like C1. We're then converting it to the p-values to be numeric because of this less than symbol. It will think that it's a character above. We're going to sort those different p-values, so it goes from the very, very smallest one all the way down to much larger ones. So there's a long kind of vector there in res2. And then we're going to correct for multiple testing there. So p.adjust takes on board that vector of p-values, uses the Benjamini-Hochberg correction approach to readjust these to a more accurate p-values given the number of tests that we have done. So if we've done 24 tests, that will mean these have been rescaled, and we'll find that these are maybe are more larger values all the way through. So what we can do is view our revised data. So you can see here we had, like before, the cell, the compound concentration, the nickname, the number of values, the mean standard deviation, and then the p-value. And what we've done here is replaced in this column is the adjusted p-value. So just slotted that in there instead of determination of whether something significant or not, because that isn't really meaningful. So you can see here we've res2 significance. So you put that into that column just for convenience so we don't have yeah. And so what you can see is the original p-values, this was 0.001. The revised one is now 0.005. And you see here this one was originally 0.025. The revised one is 0.08. This one is 0.08 originally. Now it's 0.16. And what we can do as well with this package is plot a standard curve. So this is kind of a logistic regression curve. So it's kind of an S-shaped sigmoidal type curve. And what we're going to do is just generate some data. So we're going to sort normally distributed data with a mean of 3 and a standard deviation of 3. So it's 10 data points. We're going to take these 10 values, we're going to divide them by 10 and add in more noise corresponding to 10 values again, with a mean of 0.1 and standard deviation of 0.1. And this is going to be our optical density. So that's a function of the concentration. So they should correspond, which is why we have the concentration inside there. So this is just to generate data we can play around with. We're going to compare the concentrations against the optical densities. And what we're using is this NPLR package, which contains the NPLR function, which allows us to do this kind of logistic modeling. Effectively, it's doing this sigmoidal type regression to estimate the optical density based on the concentration. And here we're using 3 parameters, which is probably a good idea for your data. And what we can do then is take this function we had before and plot it. So the things we're using here is get the X curve, get the Y curve, get X, and get X curve. So what this is taking on board is the fit1. So fit1 is sent down here. So we're using get X, get Y, and so on to plot this out. And so using the NPLR function. So what we see here is that we're taking the X data, get the X fit, then we're getting the Y fit, so that's our X and Y values. So you can see this is the X values plotted across here, and then we have the Y values plotted across here as well, and that's our optical density range and the concentration range. We have the text size as normal, the color is red, the point shape is a square. We've labeled the X and Y axes. We've got the title. We've pasted in standard curve with a normal title size. We've added a grid for ease of viewing. And then we use the lines to get the curve of X and the curve of Y. So that's the X and Y values for the lines. That's the green line we've plotted here, with a thick line and a kind of sea green color. So that's how this plot was generated. And then what we can look at is to put the optical density and concentration together as a data frame. So here we're putting this as a data frame STD together, and using the stats package to use the linear modeling function to model the concentration based on the optical density. So it's kind of Y relative to X by some parameter M plus some intercept C. So C is some value on the X-axis here. And what we're saying is that when we're trying to estimate Y based on X, the concentration, this is the optical density, that there's some slope, some complex slope, where we can use X to estimate Y. So that's why it says multiple parameters to model that slope rather than just one parameter. This is a bit simple here. And then we can get the summary of the fit. So this is detailing a linear model as if X and Y were linearly related, unlike the regression model we had before. So what this shows here, for example, is that we have our intercept here, which is actually negative. This is the standard error of the intercept, which is quite large. So our intercept is negative, but has a wide range around which it can be. T-value indicates that it has no substantial effect on Y, which is a large p-value as well. The optical density has an estimate of the slope is going to be 8.6. It is a substantial standard error. The t-value means that the optical density and the concentration are highly associated with each other, and the total R squared for the model, which is mainly associated with this linkage between the optical density and the concentration, is 8.3, which means that they're highly correlated. So then we can kind of print this linear regression model together. So what we can do is use the package ggplot2, and use the ggplot function. We're taking on that data frame, so we have to make a data frame out of this because ggplot won't work. Then we use the aesthetics function to look at the X's optical density, Y is the concentration. Replace the in title of a standard curve. We're putting on points that are red or with a bigger size. And then we're modeling our line of fit2, which we created on the previous page with the stats.lm package, to model the optical density against the fitted line with a green color and kind of black and white background theme. So that's how we get this together. So it's actually the same data we had before when we did our sigmoidal curve, and it was shaped like this. So we can fit different models to our data to see what's the best fit depending on what you're looking at. So let's now switch over to R to explore this further. Okay, so now we're in our environment where we're going to look at this bioassays package. So we have to install a few different packages for this, so including the package itself, and we can just install that. There might be a few dependencies, so just let those install. You have to activate the package like in line 13 and check the package version just as well. We also have other packages to install like TCL-TK, which lets us select the folder for our analysis. We need to activate that. We can check the package number as well, which is 4.2.2. We need to install dplyr as well for data manipulation, and we need to activate that package too. We can check the version as well, which is 1.1.1. We need ggplot2 for visualization. We need to activate that and check the package number 2. So let's just be patient while that's installing. And other packages that we use are reshape2, again for plotting as well, and then NPLR, which allows us to use that non-parametric 3-parameter model for standard curve fitting. So you can see there, ggplot is 3.4.2. And if you see things to pop up to restart, or you generally don't need to, we may need to install more recent packages that are of interest. So let's look at this package now. We can import the raw data for the 24-well plate, and what we'll see is like this. So labels A, B, C, D, 1 to 6, that matrix with the values inside there. So if we look at what's inside there, it's kind of a table. And if we look at the type of data type that it is, it's a list. So it's not quite a data frame yet. And we can do the same for the 96-well plate, and it just contains more data, 1 to 12 and A to H, and 384 is going to be A to P and 1 to 24. We can also look at the metadata as well, and import that too using the data function. So these are inbuilt functions inside our package. So what we can do is take the raw data for 24, or take the plate type, and import that into the data matrix as a data matrix variable. So we're just looking at that. And looking at the plate to data frame, we can look at that, how we convert that into a data frame for the rows and columns, the positions, and the values. We can look at the structure of that. So you can see there, each of them contains the corresponding column with those values. And this is a data frame. So it's now in a data frame format instead. If we do the same for the 96-well plate, it's pretty much the same thing. The row, the column, the position, and value. And we see there we have 12 columns now. For the 384, again, it's the same structure, row, column, position, and value, except we have 24 columns instead. If we view the values of these entries for the 96-well plate, we can do that with the matrix 96 function and view that as a table. So you can see that there. And then if we want to view the IDs for that, the metadata, we can do that as well. So taking the meta file 96 and looking at the IDs inside there. If we look at the structure of that metadata file, you can see there it has the type as well as the ID. So if we did change this to a different feature like type, we can see that being displayed. If we change that to, for example, the concentration, we can see that inside there. So it depends on what we're interested in looking at. So here we have the compound and the dilution series, we want the types, the concentration. So if we put that into the plate details, you can see that this, that deleted. Plate details, you can see that this list contains that data, and we can access that data by just putting a $sign and then selecting, say, the compound, if we want that, or the dilution or concentration or whatever we want. And this is just data we've created ourselves. If we look at how that links to the metadata file, we have the dilution, concentration, and so on. And we can merge the plate details with the metadata file by type. So you see type is inside here, SD1, S1, and so on. So if we merge them together, and now look at our plate meta variable that we've created, you can see that this data frame has the compound linked in there as well as the concentration detailed where the NAs have been changed to 0. And we have the previous information there as well. You also see that we have the all the information there. You can see the first few rows as well there too. If we do want to visualize our metadata file for the ID, in this case, we can change that, put the meta file as a matrix and use the heap plate function to visualize that. So let's try that line 78. So you can see our plate there. If we change the size to be 4, you can see the plate decreases in size. If we change the size to be 1, it gets very small. If we change the size to be 6, it'll probably be too big. Yeah, it's a bit big here. And then we can change the title to be whatever we want as well. If you change that to say be type, then that will also change. So you can see now we've got the blanks and all that information listed there. Whereas before it was just a blank sample and standard. If we change that to be concentration, you can see that they're all the same in this instance. And we can do that for the 96 mil plate for the data as well. So you can see there we have that matrix plotted for the actual value, for the values aren't shown inside there. And the same for the 384 plate, where we might need to toggle the size to be small to fit the area nicely. So another feature of this package is the reduce blank function. So the reduce blank function will take on board our raw data for the 384 plate in this case. And what we're going to do is convert that to a data frame for D in function D1. So we look at the head of D1 to see what that's doing. It's just a row called position and value. We then take our metadata file, which contains the row, column, position, which is how we're going to match them together. And then we have the values. So basically, we can think of it this way, that we're putting our values onto the meta file 384. And we're calling that D2. So that's what our inner join function is doing in dplyr. It simply adds columns from Y to X. So we're adding columns here. Don't need dplyr here. Let's do that for D2. And then look at the head of D2. So you can see there is our value added in, as well as the cell, compound, concentration, type, and dilution. We can then look further that if we view D2, you can see there we have, I can access my screen, certain ones that are blanks. So these are the blanks at the top. We have some blank 1 and blank 2, blank 3, blank 4. So what we can do next is reduce blank. So we're taking in D2, we're taking in for drugs 1, 2, 3, 4, which correspond to our compound column here as well. And then we have our blank vector, so blank 1, 2, 3, 4, which we had as well. And we're arranging this by value. So that's how we're estimating these. So if we look at now view blank, so blank minus the blank minus column is listed there. So we have our original blanks there, blanks 1, 2, 3, 4. We had original dilution, and this is the blank minus column to estimate the effect of these blanks for each of these drugs. So you can see there for drug 1, it's actually averaged out. For drug 2, it's also averaged out. Drug 3 is also averaged out, and drug 4 is also averaged out. So there's no net effect for this column. So we can view that histogram of that data for the blank noise column. So you can see that's adjusted to adjust for the effect of the kind of background, but the net effect is 0. So it should improve the accuracy of our data without changing the underlying values. So now we can take D2 we had before, given a nickname like 384 well 2, with our blanks and column names. So those column names correspond to the ones we had before. down here, and create a data frame from that and look at the structure of that. So now we have 48 rows with 9 variables. So this is summarizing our data together. So if we look at result 2, it's allocating the different rows in original data in D2, where we have multiple entries, say for compound C1. So if we look at C1, we have our entries there for C1, and it's adding these into C1 for drug 1 and the cell type as well. So you can see there we have the cell type. So you've got 7 entries here corresponding to that. When the for send for HUH7, then we have other C1s as well. So you can see those are added in there in result 2, corresponding to 7 values. You have the mean for those ones and the standard deviation as well. You see our labels added there, and then we have the treated and untreated groups too. So if we go back to D2, we can see that some are treated for drug 1 and HEP G2, some are treated for drug 1 and HUH G2, and then some are untreated as well. So that's how these correspond to the untreated 8 in here. And that goes across all the different combinations that we have present. So you can see the 3 different concentrations. The type is treated and untreated. We have the drug compounds and the cells type too. So that's quite a lot of variables linked in there together. So that then allows us to compare against a reference group. So our reference group here is C1, but it would depend on what you're looking at in your particular experiment. We're looking at the sig value is the alpha value threshold, and that's our data frame that's been sent there to this function p value, which basically performs a t-test on the data frame. So if we do this and look at the structure of res1, we'll see that this is what's produced. We have the original cell line, compound, concentration, the label, the numbers for each comparison. So that's the sum of the 2 groups. The means, the standard deviation, and then the p-value and its simple uncorrected p-value significance inferred. So we know that the significance thing is not really informative, and we know that the p-values need to be corrected, and we also know that some are controls and therefore cannot be tested. So we need to consider that in our data. And sometimes we have other symbols inside in res1 here, which we don't see here in this instance, but they might be in there as well, these less than symbols. So that's why if we look at res1.p value, we have to consider the controls or other non-numeric characters inside there. So we need to substitute these less than symbols and spaces out of there. So none are present here, but just in case. We then select the ones that have a p-value less than 1. So if we do that, we can see here, what we're doing is selecting the ones without any control inside there. So those ones were not tested, so they're not part of our analysis. That's where we get res2. So if we look at res2, you can see that we now have just 16 columns inside there. We make the p-values numeric. So if we actually look at the type of the original p-values, they're characters, so we now make them numeric. Because if you try to do a calculation on a character, it's not going to work. So now it's numeric. And then we order them. So we're going to order them together. So you can see that they're now ordered. So put that into res2. So you see res2, now they're ordered. That makes it easier to correct them, just in terms of knowing which is which. And then we're adding this into the significance column just for ease of manipulation. We're using the p.adjust function that takes on board these ordered p-values now, and it's using the Benjamini-Hochberg correction. Strictly speaking, reorder them is not necessary, but it's maybe sometimes easier to interpret the tables at the end. So if we do this now, we can view the final res2. So you can see there our revised p-values are shown. And you see that they differ somewhat from the original uncorrected p-values. And we can plot them together. So if we plot res2 daughter p-value for the original ones and res2 daughter significance for the corrected ones, you can see there that they are correlated, but you can see that the ranges differ somewhat. So we can add a grid on that if we want to view it more easily. So if they're perfectly correlated, we get a perfectly linear correlation, which is not quite what we see here. Okay, so let's look at the plotting a standard curve. So this uses the NPLR function, and we're creating some data here in concentration. Just some data to work with. You might have your own data, and here we're using a range from about 0 to 10 for our data. The optical density is based on that, but it's scaled to be a smaller range of values. So if we look at the OD vector, you can see there it's going from 0 to 1, really. And then we look at the fit between concentration and the optical density for a 3-parameter model without using a log-scaled data here. So we can look at fit1 in this case, and then plot it down here. So we're using the getX function to plot the values and the Y values to get the Y values using a square point size, normal text size, red points. The Y lab, the X lab is labeled with concentration. Y is the mean optical density. You can see there, and then we have the standard curve as the title with normal text size and a grid placed on top. If we use logs and just turn this to true, what you find is that that data is kind of shaped differently because it's now log scaled. And I'm not sure for this data that we need to do that because there's already a decent spread on it. So it doesn't really make sense to do that for this data because it's less easy to interpret. If we move beyond a 3-parameter model, so let's look at the original, the getX curve, get Y curve, you can see there, it's fitted there. If we use 4 parameters, it kind of gets over parameterizing maybe, in terms of trying to fit the model that you have here. But you can explore that more with the NPLR function. I note that each time you run this, it will recreate different data. So you'll find that the curve is actually shaped differently. So you can see that there on the right-hand side. So let's look at the linear regression. So here we have to create a STD data frame. And we're using the stats function as a linear model. So remember, we're linearly modeling it as y tilde x. So we're inferring, trying to infer optical density, look at the association of that with the concentration. Let's apply that and let's check the summary of our linear model. So what we can see there is the optical density is the strong effect. And let's look at the plot in the ggplot. So that's, we're plotting here the data frame of the optical density and the concentration for a standard curve with the red point size of a particular size using the fitting a line using fit2, and we're calling it C green. So what we can see here is for this plot, the concentration optical density axes have been flipped around. So it may make sense to match our previous one up above to switch them. And to do that, we just need to change the corresponding X and Y values here. So we need to switch around this formula for concentration and optical density for fit2. So you can see there, we still have an R squared that is very high, it's 0.91, which is the same as what we had before. We're just rearranging our data. I'm going to plot that again with the revised axes. You see concentrations on the bottom, optical densities on the top. You can see that this shape is different from the one that we had before with the NPLR's functions, which was sigmoidal. So this kind of shows how we can do different types of regression, linear or logistic, on our optical density data. So you can see that this kind of shows the different things we can do with bioassays in terms of plotting different plate data, visualizing the values, visualizing the metadata, linking them together, performing some basic statistical testing, and then sort of inferring data using different regression models for our standard curves. So let's discuss any topics you want to discuss in the labs themselves.